Hello everyone, welcome to my YouTube channel. So if you're new to this channel, kindly subscribe for more videos like this. So in this video, we are going to look at biology paper 2. Interesting question, which may even come in your exam. So the question is saying figure 8.1 shows a cell from the Palisade layer of a leaf. So as you all know what to say, a leaf is an example of an organ in plants, as well as a stem is an example of an organ and a root is an example of an organ. Maybe in an exam they can ask you to say, list 3 examples of organs in plants. So you are able to mention those. So we have a palisade cell which is found in the leaf. So how sometimes they can bring a cell like this one, and then they are also going to ask you to identify the same cell. So you're able to identify a palisade cell because of the abundance chloroplast or chlorophyll. So you all know to say that this structure one is called the chloroplast or chlorophyll, if you want, whichever way you want. So it's a chloroplast or chlorophyll. So in the chloroplast or chlorophyll, that is where now the process of photosynthesis takes place. And one thing that you need to know is that the chloroplast is only found in plants. It is not found in animals. Then structure 2 is called the vacuum. So the vacuum is made up of cell sap. Okay, it's made up of cell sap, and cell sap is just a mixture of salt, sugar, and water. So it's more like a solution and water. So let me just say H2O. So it's a solution or a concentrated solution. So that is its function, containing cell sap, which is a solution. Then for structure 3, this one is called the cytoplasm. So cytoplasm is a jelly-like fluid. So this is where now the site, it's more like a site. So when I use the term site, I just mean the place. It's a place for chemical reactions. So for cytoplasm, it is present in both plants and animals, but when it comes to the vacuum, it is only present in plants. So that's one thing that you need to know. What about part 4? Part 4 is the cell wall. Okay, a rigid structure. So the cell wall is only present in plants. It is not present in animals. It is also present in bacteria, like you talk of the prokaryotics. Then we have structure 5, which is called the cell wall membrane. Okay, so we have structure 5, which is called the cell membrane. So what is the function? What is the function of the cell membrane? So this cell membrane acts as the selectively semi-permeable membrane. That means it selects what goes in and out of the cell. So that is its function. And then structure 6, this one is called the nucleus. So the function of the nucleus is to store DNA. That is store DNA, which is deoxyribonucleic acid. That is DNA. It also controls the cell activities. All the cell's activities are controlled by the nucleus. So now let's try to answer the questions here. So we have this question which is saying in table 8.1, check the numbers that label the 3 features of the palisade cell, which are also present in animals. So one thing that you need to know here is this: the cell wall is not present in animals, the chloroplast is not present in animals, as well as the vacuum is not present in animals. Of course, it is present now in small amounts, so it is insignificant. So these are the structures which are not present in animals. Now, this question is asking us to label the 3 features which are present in both, or which are also present in animals. So what are those? We have number 1, the cell membrane. We also have the nucleus, as well as the cytoplasm. So you just have to tick. Okay, so for cytoplasm, it is represented by 3, so I'm going to tick this one. For the cell membrane, it is represented by 5, so I have to tick this one. And then the nucleus is represented by 6, so I have to tick this one. So we are done with that question. So for the second question, is saying state and describe the function of 2 features of the palisade cell that are only found in plants. So you just have to choose. Say it's a free question that you have to choose. So from here you have to choose 2. You can talk of the vacuum, you can also talk of the chloroplast, or you can talk of the cell or whichever cells, the cell structures that you want. So let me start with the chloroplast. So after naming this one as chloroplast, I have to say this is chloroplast, isn't it? Or if you want, you can just say chloroplast is represented by which one. Don't know if it's 1, you say 1. Then you explain the function of the same structure, which is 1, which is the chloroplast. So the function of the chloroplast, we can say site of photosynthesis. Site for photosynthesis. So you know to say that during photosynthesis, what is produced is what? Oxygen, and energy is produced. Then another one, I can choose cell wall or vacuum. Okay, so I'm going to choose the vacuum. So the function of the vacuum, like I said the function of the vacuum, it stores cells up, or it contains cells up. So you can say contain cells up. So I explained what a cell sap is. It's more like a solution which is made up of sugar, salt, and as well as water. So contain cell sap. Then let's see other questions. So question C is saying figure 8.2 shows some red blood cells, which are animal cells. Okay. Then this question is saying which feature normally present in an animal cell is absent from a red blood cell. I think this one is an important question that you need to understand and know. We know to say that animal cells, they have nucleus as well as plant cells, isn't it? Now here we're focusing much on animals. They have a nucleus. This is an animal cell. Of course, a red blood cell is an animal cell, but it lacks what? Nucleus. It doesn't have nucleus. And this is important to create the large surface area for the carrying of hemoglobin. So it's very, very important. So the red blood cell lacks nucleus. So that is the answer. The red blood cell doesn't have the nucleus, and this is the mature red blood cell. So in top of immature red blood cell, they have the nucleus, but as it is growing, or when it becomes mature, the nucleus disappears in order to create the large surface area for carrying hemoglobin as well as oxygen. Okay, so let's see the second question, which is saying state the function of a red blood cell and describe one way in which the red blood cell is adapted to carry out its function. Okay. So in terms of the function, you know to say that the red blood cells, they carry oxygen around the body, or they distribute oxygen to the tissues of the body. Apart from distributing oxygen to the tissues of the body, they also carry small amount of carbon dioxide from the tissues to the lungs. So those are the functions. Just have to choose one. Apart from that one, you can also talk of, you know to say that this is the outer part of the red blood cell. This one is called the cell membrane, isn't it? So on the same cell membrane, that is where now you are going to find the antigens. So the antigens are present on the surface cell membrane of the red blood cells. So you just have to choose one. So transport oxygen from the lungs, transport oxygen from the lungs, to the tissues. Okay, to the tissues. Then from there you are done. If you want, you can also include another one. Also, the cell membrane contain the antigens. Cell membrane contain antigens and genes. Then from there, if you want, you can also say transport, but you just have to choose one. Transport carbon dioxide, or transport small amount of carbon dioxide, from tissues to the lungs. Then this question is having two questions, and also we have to talk about the adaptation. So you talk about adaptation, you're looking at the structures that the red blood cell has for it to perform its function. Like for example, it lacks the nucleus. It also has the biconcave in shape. So the biconcave in shape is the structure that you are able to see here. This is the biconcave in shape, this one here. So this biconcave in shape, it enables now this red blood cell to carry hemoglobin. When you talk about biconcave, you just need to imagine it in this form, like the way the plate is. The plate is able to hold soup because it is biconcave. That's the way the red blood cells are. So they are biconcave in shape, and this biconcave in shape, it enables it to carry hemoglobin as well as oxygen. So you just have to put that one. Okay, let me just bring you back to Same question, yes, this one. So we are going to say presence of biconcave in shape, presence of biconcave, concave shape to carry hemoglobin, or to carry more hemoglobin, then you're done. Then apart from that one, I also have another question here. Just see, yes. So this one is coming from dentition as well as digestion. In fact, just focusing much on digestion. So this one is saying teeth are involved in mechanical digestion. What is meant by the term mechanical digestion? So in talk of digestion is just the process by which now the food is broken down into smaller particles. Okay, now this digestion can be mechanical, which is also known as physical, which is done by the teeth. Then also have chemical, which is done by the enzymes. So now when you talk of chemical digestion, it's the breaking down of large food particles into smaller pieces by the use of teeth. So you can write that one. Is the process, okay, the process of breaking down large particles of food, into smaller pieces, pieces by use of teeth, by the use of teeth. Then from there you are done. So that is the way you're supposed to write that one. Then the second question is saying figure 7.1 shows a section through a molar tooth. So we have different types of tooth. So we have what we call the canine, we have what we call the incisors. Also have what we call the molar as well as the premolar. So these are the types of teeth that we have. So in talk of the structure of the incisors looks something like this. So it's not going to be perfect. I'm just going to try to label it time. So this is the way the incisors looks like. So this is the part which is the crown. This is the root part. It's made up of the enamel and other structures. But when you talk of the incisors look something like this. Okay. Then it goes like that. So this is the way the incisors looks like. Then when you talk of the molar is the one now which is in this diagram here, and it has two roots, as you can see. So these other tooth, like the canine, the incisors, they just have one root. So these are the roots which I'm talking about, these ones here. So for the premolar, it looks like the way the molar is, but the only difference is that for the premolar, it just have one root. So it is going to look something like this. You draw like that. So it's going to look something like this, and then you draw all other structures. But the only thing which is going to be different here is that it is just going to contain one root, as you can see here. Okay. So in terms of the functions, this one is used for cutting, this one for biting, this one for grinding, as well as chewing. Then also the premolar, it has the same function as the molar. Okay. So the question is saying on figure 7.1, label parts Q and R. So for part Q, since this one is the enamel and this one is the pulpy cavity, the part which is in between the pulpy cavity and the enamel, so the enamel is the hardest part of the tooth and it is the outer part. This one is called the dentin. So the dentin is the one which is sensitive to any change, or it's responsible for sensation as well as pain. This is because this is where now the nerves attaches. You know, to say that the nerves can send the electrical impulses when the receptors are exposed to the stimulus. All right. Then for structure R, this one is going to be the pulpy cavity. The pulpy cavity. So pulpy cavity contains the blood vessels as well as the nerves. Okay, so the second question is saying state 2 reasons why this tooth can't be a canine tooth. Okay, so like I was drawing those structures, the canine, the incisors, premolar. So you saw what was happening. For the canine, it has only one root, but when it comes to this premolar, it has 2 roots. So we are going to say molar has 2 roots. Has 2 roots, while canine has 1. So has 1 root. Then apart from that one, we can also talk about the structure of the enamel, like the way the incisors was looking like. It was looking like this. Okay, let me just draw this one just rough sketch of a canine. So this is the structure of the canine. As you can see, the way the enamel is structured, it is very small compared to the molar. So we can say the canine has small. The canine has small enamel, has small structured enamel, while for the molar it has bigger enamel. For the molar it has large enamel. Okay, large enamel. So those are the differences that you need to include. So for the second question, for Roman number number 3, saying gum disease causes the gums to shrink from position T to position S, as shown on figure 7.1. So let's see. So we have position T, we also have position S. Now, that is where the gum disease are likely to occur. Now the question is saying, suggest why the tooth is more likely to decay when the gums are at position S. So in terms of the way the tooth is divided, we have this one here, which is the crown, and this one here, which is the neck. Let me say this one is the neck, this part here is the neck, and the entire part here is going to be the root. So the way it is, if the neck is exposed or the root is exposed to the external environment, to say that the GIT is not part of the intracellular structure. It is an external part, because the GIT is a continuous tube from the mouth up to the anus. So the contents in the GIT are not part of the intracellular. So one thing that you need to know here is that if the position T, or if the neck comes to position T, meaning to say the root is going to be exposed to the internal environment, and to say that the bacteria, they are able to invade in gaps. And once they invade in gaps, what is going to happen is that there, if the food stuck in those gaps, the bacteria, they are going to catabolize those food, and they are going to produce an acid. As a result, you find that this enamel will be hydrolyzed. Okay, so the reason why it is likely to occur at that point, this is because we can say this is because the roots are exposed to the environment. Roots are exposed to the environment, are exposed to the environment. Okay, so environment. Then apart from that one, once there is a space here, let's go back to this point. Once there is a space here, you find that the food is going to stack in spaces. You're going to agree with me that if you find that you are having some spaces in your tooth, the food will stack, isn't it? And what is going to happen is that once the food stack, the bacteria or the bacterium, they are going to metabolize those food particles or the residues and produce acid which is going to neutralize or dissolve this enamel part. Okay, so we can talk of we're going to say food stuck in the gaps, food stuck in gaps are hydrolyzed by bacteria, or let me say not bacteria but bacteria or bacterium. There is no way that bacteria is hydrolyzed by bacteria and produce acid. Produce acid. So this is acid. This acid is very dangerous in that it is going to dissolve the what? The enamel. Then for the fourth one, say state 2 ways of maintaining health teeth. So when it comes to maintaining the health teeth, you have to brush twice a day. So we can say brushing teeth properly twice a day. Properly twice a day. Then apart from that one, we can talk of eating friendly food by avoiding sweet foods like those that contain sugar. So you can say by eating friendly food. Okay. Or apart from by eating friendly food, you can talk of or avoiding, or we can say avoiding sweet foods or sweet food. Sweet, not sweet, but sweet food. And others, or by visiting the dentist regularly. You can also include that one. So we can say by visiting the dentist regularly. So these are the answers for this question. So let's see, we still have more questions here to come. So this one is coming from excretion, okay, which is saying figure 5.1 shows a close section of a kidney. So you have structures G, E and F. So when it comes to the kidney is divided into two. We have what we call the cortex. We also have what we call the medulla. So in terms of the structures, we have this part here, which is in gray. It is called the lenocortex. Then this part here, the white part, and also these lines which are showing the nephron or the nephrons, this part is called the medulla. Okay, it's called the medulla or medullary. Then we have part G. So this part G, it is the ureter, but since it is near the kidney, you can just say this one is going to be the lenopelvis. So there's a difference between the ureter and the lenopelvis. So we can say if we cut from here, this one is going to be the lenopelvis, and this one is going to be the ureter going forward. So this one can be the lenopelvis. Okay. So one thing that you need to know is that the functional structure unit of the kidney is just the nephron, the one which is involved in filtration of substances and the absorption of substances as well as secretion. So name the structures labeled E, F and G as shown in figure 5.1. So we have E. So E is called the lenocortex, or we can say the cortex if you want. We have the lenocortex, and then F is called the medulla. Then for G is called the lenopelvis. So those are the answers. Then from there, we have this question which is saying explain the function of the lenocapsule in the kidney. So in terms of the structure of the lenocapsule, where are we going to find the lenocapsule? So the lenocapsule is just the outer part here. This is the lenocapsule. So this lenocapsule is the outer part or the outer structure, and its function is to protect the kidney, or to protect the internal structure of the kidney from the external environment. That's its function. Apart from that one, it acts as a barrier between the outsider and also the inner cells of the kidney. So from there we can say it protect and act as a barrier. Protect and act as a barrier. And act as a barrier. Okay, so those are the functions, or that is the function for the renal capsule. Okay. The second one is saying glucose is absorbed back into the blood by active transport. Define the term active transport. So what is active transport? So one thing that you need to know is that active transport, there is the use of ATP, adenosine triphosphate, ATP, that is going to provide energy for the movement of particles. So when you talk of active transport, is the movement of particles from the region of higher concentration to the region of lower concentration against the concentration gradient by using ATP. So I'm saying E is the movement of particles. Movement of particles from the region of higher concentration. Let me say from the region of higher concentration to the region of lower concentration. So this one has to be higher concentration to the region of lower concentration against concentration gradient. Against the concentration gradient. By using ATP. By using ATP as energy. Or you can say by using energy if you want. That is active transport. For example, the reabsorption of glucose needs energy from ATP through active transport. Then the second question is saying give one example other than glucose of a substance that is reabsorbed into the blood from the lumen or from the renal tubule. So we can talk of calcium, potassium. So potassium, calcium, sodium, all those, and chlorides, they are reabsorbed. If you want, you can also talk of urea. Yeah, they are reabsorbed. Now, let's move on to the next question. This one is saying dialysis is a treatment for kidney disease. Figure 5.2 shows a dialysis machine. So we have a dialysis machine here. So let's do a review of this dialysis machine. So we have this part here. So this tube here is connected from this tube is connected from where? From the artery, isn't it? It's connected from the artery. Then it's moving that side. Then this is the vein. So this is the artery. And this is the vein. Okay. So the way the dialysis work is like this. It is filtering, or it filters toxins and substances which are not needed by the body, like urea, excess water, as well as salts. So as you can see here, we have two parts. We have the used dialysis and flash dialysis. So in the flash dialysis, what are you supposed to find in the flash dialysis? You know to say that in the blood, you have to find glucose. It's a must in blood. You have to find urea. It's a must. You have to find salts or salt. You have to find excess water. It's a must. Now, when it comes to a dialysis machine, this one here, it helps to filter substance by diffusion and other by osmosis. Two processes, diffusion as well as osmosis. So this is the flash dialysis. So in this flash dialysis, there must be substances which are going to be equal or which are going to be in low amount compared to the substances in blood. Like for example, when you talk of glucose, so the first thing that they are going to do, they are going to measure your blood glucose. So if they find that your blood glucose is somewhere 3.9 or 5.9 moles per deciliter, they are going to make sure that the amount of glucose in blood, it has to be equal to the amount of glucose they are going to add in the flash dialysis. This is because when there is equal amount of glucose in blood as well as the one which is in dialysis, you're going to find that there will be no net movement. This is because glucose is an important substance to the body, which is needed for respiration. So here, the glucose in the dialysis tube here, this one is called the dialysis tube. It has to be equal to the glucose outside the dialysis tube, so that there is no net movement. Apart from that one, urea is contained in blood, so it is in the dialysis tube. That one, it has to be in high quantity in the dialysis tube, but outside the dialysis tube, you need to make sure that there should be no urea, so that you excrete urea by diffusion. Then when you talk of salts, of course, even salts, they are high in the dialysis tube here, but here there should be no salts, as well as excess water. Here there should be fewer water, and here there should be excess water, so that they should be filtered out. Now, you find that since this one is the flash dialysis and then this one is the used dialysis, so in this used dialysis, after filtration of substances has occurred in this dialysis machine, you find that in this used dialysis fluid, there will be high urea. So you are going to have high urea because it has been filtered out. So high urea. Then apart from high urea, in this one you are going to find that there will be high salts as well as excess water. There will be excess water in this used dialysis fluid. But the only thing which is going to be different is that since glucose is not reabsorbed, is not secreted into, or is not filtered, so here there will be no glucose in the used dialysis. That is one thing that you need to know here. So now let's try to answer the questions that follows. So I have this. Okay, let me just display it. So this one is saying the composition of the dialysis fluid changes as it passes through the dialysis machine. Complete table 5.1 using the words low, high, or same, or none, to show how the concentration of each substance changes in the dialysis fluid. So the last one has been done for you. So in terms of the toxins, they are going to be high in the used dialysis fluid, which is true. What about urea? I said it is going to be high. Okay, so even the blood before dialysis, it's going to be high. What about salts? Salts are going to be high. But when it comes to the fluid used in the dialysis fluid, you are not going to find glucose there, so none. But what about the flash dialysis fluid? There has to be the same. Okay, so you're going to say same. So when I say same, so meaning to say the one which is in the dialysis tube and the one which is in the flash dialysis fluid, it has to be same to prevent the filtration of glucose. This is because it is very important substance which is needed for respiration. Then when it comes the flash dialysis, in terms of salt, we can say none. Then when it comes to the urea, we can also say none. Okay, or you can say low or none. Then the second one is saying explain how the dialysis filters blood. So here they are asking us to explain how the dialysis filters blood. So we are going to say the dialysis machine acts as the artificial kidney. So a person is using dialysis, meaning to say the kidneys are not working. So since the kidney are not working, meaning to say the person is failing to filter toxins and other substances like urea, salt, and excess water. So if a person is failing to filter all those substances, so meaning to say it's going to use the dialysis machine, which is going to act as artificial kidney. So we can say the dialysis machine acts as the artificial kidney by filtering blood. Okay, by filtering toxins, dialysis machine act as the artificial kidney. Artificial kidney by filtering toxins, urea, salts, so this is air salts, and excess water. And excess water. Blood is not filtered out. So I'm going to say blood is not filtered out because when you talk of the dialysis tube, that dialysis tube is selectively semi-permeable membrane. So it has small holes. Since blood is made up of large particles, it is not filtered. Blood is not filtered out. These are filtered out because it has large particles, or it is just big to close the dialysis tube. This is because we can say it is big to close. It is big to close the dialysis tube. Then from there now, we can say glucose is not filtered out also. This is because it is an important substance needed for respiration. So you find that the concentration are going to be the same, which is the concentration inside the dialysis tube as well as the concentration outside the dialysis tube. They are going to be the same. So you can say glucose is not filtered out also. But of course, yeah, can include that one. But this question was just focusing on how the blood is filtered. So glucose is not filtered. Out. Because the concentration inside the dialysis tube, inside the dialysis tube is same to the solution outside. Solution outside. So there is no net movement. So you can say, so there is no net movement. Then from there now we can say that's the end of this one here. So there is no net movement. Now, the second one comes by saying kidney transplants are the most common organ transplants. Describe the advantages of a kidney transplant compared with the dialysis. So when you're using dialysis, meaning to say you'll be visiting the hospital regularly, like 2 days in a week, you see, but when you undergo kidney transplant, meaning to say that one act as what? The normal kidney. Okay? So meaning to say the person will live a normal life. So the person will live a normal life. Fine? Person will live a normal life. So when a person undergo kidney transplant, you find that the person won't visiting the hospital regularly. Yeah. Then apart from that one, the kidney are just going to filter out the toxins, normally like the way the normal kidney is. Once the person undergo kidney transplant. So we are going to say the kidney transplant operates as the normal kidney and also prevent, but this one makes a person not visiting not visiting the hospital regularly for checkup or for dialysis. Makes a person not visiting the hospital regularly for dialysis, visiting the hospital for dialysis, which is important, isn't it? Yes. And also, you find that when you are using dialysis, it's going to be more expensive throughout your life than when you go for a kidney transplant. Of course, a kidney transplant may seem to be very expensive at first, but once it is done, you find that you never spend any money to do the filtering. Okay, so it is cheap in that way. So all those are answers. You just have to answer them like that. Okay, so let's see the last question. Yeah, the last question. This is also an important part that you need to understand. So it is saying figure 6.1 shows the changes in glucose concentration of the blood. So we have high glucose concentration here and low glucose concentration here. So name the process that maintains blood glucose concentration within set limits. So this one is called homeostasis, because you need to say that homeostasis is the process of maintaining the internal constant environment. When there is a change, you maintain. Okay, so in this case here, we're going to say homeostasis. Let me say homeostasis. This is E. Homeostasis. This is S and this is S. And just write it nice so that you are able to see. Not like this. So homeostasis. Homeostasis. That's the answer for that one. Then name the hormone that would be secreted in response to the increase blood glucose concentration at A. So as you can see here, there is an increase in concentration of glucose here at A. So now which hormone is going to be secreted? Of course, it's insulin, the one which is going to bring the blood sugar level into normal. So that is the insulin. Then name an organ that is responsible for the decrease in blood glucose concentration at B in figure 6.1. So at B there, what organ is that one? Just the liver, isn't it? So you can say the liver. So insulin converts glucose into liver glycogen as well as muscle glycogen. Okay, so name the compound that is converted to glucose at C. So as you can see here, there is low level glucose. So when there is low level glucose, you find that glucagon is going to be secreted, and once glucagon is secreted, that one is going to convert liver glycogen into glucose. So the one which is secreted here, the compound which is secreted to convert glucose into C, or at C, that one is the glucagon. Glucagon, which is also the hormone. So this hormone is secreted by the alpha cells of the pancreas. The insulin is secreted by the beta cells of the pancreas. So that's all for this video. Remember to subscribe if you find it useful. You can also share it with your friends.